In this video, we will discuss the third topic of our present series, which would be about proteins. Now, before we continue with our discussion, let's ask a question. What do you need to take out the energy from this particular food? Now, the answer to this is very familiar with us. Of course, we will be saying, in order to take out the energy from this food, we have to put it in our mouth, chew it, and then our digestive system will eventually digest it. So in order for us to digest, what do we actually need? Well, we essentially need enzymes. So for the energy to be taken out from all the food through digestion, particularly this part of process, we need what we call as enzymes. And that would be what our topic is all about. So for module 1.3 about enzymes, our objective would be number one, to describe what is an enzyme. So we must understand its description and its definition. Number two, give examples of enzymes and why they are very important in the human body. So we'll look at some examples and how they function in our body. And number three, discuss how enzymes lower the energy of activation of the chemical reaction they speed up. So you will be noticing in this objective that the major function of the enzyme to speed up, and we'll be looking at the mechanism behind that. With that, enzymes can be defined as proteins that act as catalysts within living cells. To further define that, enzymes are protein catalysts that increase the velocity, increase the speed of a chemical reaction, and are not consumed during the reaction they catalyze. So you will be seeing here two important parts or description about enzymes. Number one, its main function is to increase the speed of chemical reaction. And number two describes what happens to it. Nothing happens to it during its job as an enzyme because it is not consumed during the chemical reaction or during the reaction. So let's look at some important information about the enzyme. Our SLG mentions that the enzymes are naturally produced in the body, and then it is also very important for performing certain tasks such as muscle building, DNA synthesis, gene expression, digestion, as mentioned a while ago, and several other important functions in our body, important reactions that happen in our body. So you can see as early as now that enzymes are indeed so important for living organisms. So let's talk about mechanism of enzyme activity. So how does the enzyme do its job to speed up the chemical reaction? Well, first of all, you must have the enzyme present, and of course there will be a substrate that will be present. So what is a substrate? Simply stated, it is a ligand, a molecule wherein this will be binding with the enzyme, producing an enzyme substrate complex. And because of their particular orientation and because of the formation of the enzyme substrate complex, you will then produce products. Whether the products would be putting them together or separating them into smaller parts or smaller pieces or smaller bits, that would be the mechanism of enzyme activity. So again, the substrate or the ligand will be, or called substrates, will be converted rapidly into different chemically modified molecules, and those will be called as products, while the enzyme remains unchanged during the whole entire process. We'll be talking about later on the actual process that causes the substrate to become products, but as early as now, or at least for now, we know that the substrate must bind with the enzyme to produce the enzyme substrate complex, and then later on, because of certain reactions, products will be produced after that. Now please do take note, as we have discussed in the previous discussion, that the conformation of the enzyme is very important. That's why we talked about the primary, the secondary, the tertiary, and the quaternary structures of levels of structures of proteins. Now, take note that the ability of the enzymes to catalyze the reaction would be dependent on the native conformation. So, if they will lose this native conformation, they also lose their catalytic activity. In other words, if the protein is denatured, it will lose its ability to speed up chemical reactions. That is what they are supposed to be doing. That is why very important that their native conformation should remain untouched. So we have already discussed this last time that normal protein would look like this one, but because of extreme environment, such as extreme temperature, extreme pH, which would be disrupting the shape of the protein, this will be resulting to a denatured protein wherein more or less the protein will become unraveled, and now without the three-dimensional conformation it will not be able to do its job as an enzyme to speed up chemical reaction. And we're very familiar with some of the factors like temperature, like pH, substrate concentration, and enzyme concentration. So when there is an increase in temperature, the hydrogen bonds will be disrupted, which would be releasing the three-dimensional conformation of the protein, particularly in our example, the enzyme. Extreme pH, hydrogen ions will also disrupt bonds between amino acid, substrate concentration. So when you increase the substrate concentration, definitely you will also be increasing the level of reaction because of the increased amount of available substrates to be catalyzed. And enzyme concentration as well. Well, if you increase the amount of enzymes, this means that many more reaction can happen, which also increases the enzyme action. So these are a few of those things that would be factors that can affect the enzyme. Particularly, if you denature the protein, which would be the enzyme, definitely they will not be able to do their job. So, one more thing that we have to take note of, without enzymes, many bodily processes would be affected, which means life cannot exist at all on this planet Earth if the enzymes are also absent. Now, let's now go to another question. What are the 6 classes of enzymes? And this is very important for us to remember. We can remember this using the mnemonics OTHLIL, and these are the 6 classes of enzymes. O is oxidoreductase, TH or T is transferases, H would be hydrolases, L would be lyases, and I isomerases, and L ligases. So those are the 6 classes of enzyme given by the mnemonic OSLIL. So that will make it easier for you to remember these 6 classes. Now, of course, we don't just need to memorize the names. We must also be able to know what would be their description, the process that they catalyze. So in this case, oxidoreductase, given by its name, it does the transfer of electrons, which would be the oxidation reduction reaction. So that would be oxidoreductase. For transferases, as you can see already by the name itself, it would be transferring, but in this case, group transfer reactions will be done by transferases. Hydrolases, you know this already. It does hydrolysis reaction and transfer of functional group to water. Lyases functions as the following. It cleaves the CC bond, the CO bond, the CN bond, or other bonds by elimination, leaving double bonds or rings or addition of groups to double rings. So very important to remember that they not only cleave these bonds, but they also produce double bonds or rings in addition to groups to double bonds. Isomerases, very familiar already, it would be the transfer of groups within the molecules to yield isomeric form. So you're very familiar with the definition of isomers, which be having the same number of atoms, but they have different forms. So it's possible that the OH can be found on the left side, while the isomer, another form of it, can be found on the left side. So isomerases transfers these molecules to yield isomeric forms. Ligases would be the formation of CC bonds, CS bond, CO bond, CN bonds by condensation reaction coupled to cleavage of ATP or similar cofactors. So these are the these are the classes of enzymes and the type of reaction they catalyze. So it's very important for you to memorize this. Okay, here's another question. What enzymes and their functions do you know? So this is answering objective number 2, wherein we should be able to remember the names and the functions of enzyme that occurs in our body. So let's look at this one. When we talk about salivary amylase, can be found in the salivary glands. So the action or the function of salivary amylase would be to begin carbohydrate digestion by breaking down starch and glycogen to disaccharides. So that will be starting in the mouth. The amylase will digest carbohydrates. How about pepsin? Pepsin is found, or the source of it, which is gastric glands, and from the name itself, it would be beginning protein digestion. So you can see here peptide, so pepsin, and it begins protein digestion. Pancreatic amylase, of course, as the name itself implies, it sources pancreas, and the amylase will give us the idea that it will be what? Breaking down of starch, carbohydrates, particularly starch and glycogen, into disaccharides. That will be pancreatic amylase. Pancreatic lipase, from the name itself, you can already know that the source would be the pancreas, but at the same time, the word lipase, what does it do? The digestive action would be breaking down fats into fatty acids and glycerol. As you can see, lipase coming from the word lipids, so it would be digesting lipids or fats into fatty acids and glycerol. How about trypsin, chymotrypsin, carboxypeptidase? So you can actually know peptidase, carboxypeptidase. So you got an idea here. So they are proteolytic enzyme from the name proteo. You will know that this would be functioning as breaking down or breaking down proteins or partially digested proteins into peptides. And they can be found in the pancreas and the mucosal cells. How about intestinal enzyme peptidase? From the name itself, it's you know that its function to break down peptides into amino acids and found in the mucosal cell. And sucrase, maltase, and lactase, you've already probably encountered this beforehand, and the name itself will give you the idea that it would be digesting, breaking down disaccharides into monosaccharides. So lactose, maltose, and sucrose will be digested by lactase, maltase, and sucrase. So all of them are disaccharides, so they will be broken down to monosaccharides. Intestinal lipase, again from the name itself, it will be breaking down fats into fatty acids and glycerol. And those are some example of enzymes in our digestive system that would be very important to break down, digest different important molecules to produce energy. Okay, now let's now talk about the actual mechanism of how the enzyme is able to speed up. Now, given this picture, you already know that this is the conversion of reactants to the products graph. Notice that the reactants is on the left side and will become products on the right side. But also notice that the amount of energy of the reactants would be high compared with the amount of energy after the products has been produced, because the reactants would be higher in this graph. The amount of free energy is higher compared to the products which are down below here, which has a lower amount of free energy. But one thing also you have to take note of is what we call as a transition state. So this is some kind of a barrier that the reactants need to overcome in order for the process of the reactants to become the product. So you will notice here that reaction A and B must absorb enough energy from the surrounding to reach the unstable transition state where bonds can break. So that is called the transition state, and this is the hurdle that the reaction must overcome in order for the products to be produced. Okay, now take note that, as we have mentioned in the beginning, enzymes functions to lower the activation energy in order for the reaction to proceed faster compared to its natural state. So you will see in figure number 5, enzymes decrease the activation energy required for a given chemical reaction to occur. So if the amount of energy or activation energy is up here, somehow the enzyme will lower the activation required so that it becomes faster and easier to overcome and easier to produce the products. And that would be the basic idea of how enzymes are able to speed up chemical reaction. Particularly noted here is that the substrate must enter the active site of the enzyme, in order for the activation energy to be reduced so that the the reaction can proceed faster as compared to to normal conditions. So take note that the active site is a region of the enzyme that corresponds to the contours of the reactant molecules or the substrate that it acts upon. So notice that the active site has similar conformation as the substrate. This makes sure that only the substrate would be coming inside of the active site, and only this will proceed, or only this will cause the chemical, the reaction to proceed in order to produce your products. That is why, if you remember a while ago, denaturing the enzyme will cause the enzyme to lose the conformation of the active site, which also means that it will not be able to do enzyme activity catalysis anymore, because it will no longer be in conformation with the substrate. So no shape, no similar shape of the substrate, no catalysis will happen here. So that's something that's very important. And that gives us the idea like a lock and key, where the active site is here and the substrate will be exactly or almost exactly the same as the shape of the active site. And this will cause the catalysis or the enzyme reaction to proceed faster. Okay, so here are some things we have to take note of about the activation site. The activation energy for the conversion of the substrates into products is significantly lowered down, as mentioned a while ago. The enzyme binds to the substrate in such a way that would ensure that the transition state can be easily reached. And finally, there are various mechanisms used by the enzyme to lower the activation energy and speed up the reaction. Some would say that the the activation energy or the reaction follows a different or alternate pathway, and that can be true as well. So the basic idea is there are several mechanisms by which the lowering of the activation can happen. It can follow a different pathway, alternate pathway, or several other things that could possibly happen. So let's have a concept check. Why do enzymes act only on very specific substrate? What accounts for this specificity? We have already answered it a while ago. Only the specific substrate will fit properly into the active site of an enzyme, the part of the enzyme that carries out catalysis. And that's why we recall a while ago about the lock and key complex, or the lock and key theory, wherein the substrate will be exactly the same as the conformation of the active site. Okay. So this is again the enzyme activity, and just a further picture of what happens during how enzymes are able to lower the transition state, or lower the activation energy. So again, the substrate enters the active site, and then substrate are held in active site by weak interactions. Then the substrate are converted to products and released, and then the enzyme are now available for new substrate to go to it. And that ends our discussion.